Hello and welcome everyone to today's webinar where we will be discussing creatine and creatinine testing in dietary supplements. My name is Jennifer Caveney and I will be facilitating today's presentation. All right, let's go ahead and get to the presentation. Our presenter today is Dr. Łukasz Wacławik, and he serves as Director of Scientific Strategy for Contaminants at Eurofins Food Chemistry Testing, Madison. He leads the development of contaminant and residue analysis programs to safeguard food quality and consumer safety. Prior to this role, he contributed to regulatory science and research at the FDA Center for Food Safety and Applied Nutrition. Widely recognized for his expertise in chromatographic and mass spectrometric techniques, Dr. Wacławik is a sought-after speaker at international scientific conferences and has published over 40 peer-reviewed papers focused on chemical contaminants, residues, and adulterants in food. He holds a PhD in food chemistry and analysis from the University of Chemistry and Technology in Prague. Łukasz, please begin whenever you're ready.
Thank you for the introduction and welcome everybody. I'm really excited to talk about creatine and creatinine testing. Let's take a look at the agenda we have. For this webinar, we're going to take a look and explore the background on creatine and its function in the human body, which is closely related to its use in dietary supplements and also popularity of these products. And I'll also touch on its main degradation product, creatinine. We'll discuss different forms and derivatives of creatinine used as ingredients in dietary supplements. And next, I'll highlight some challenges that are faced during determination of creatine and creatinine, specifically when testing via high-performance liquid chromatography with UV detection. We'll go over internal Eurofins method and the validation data, and we'll also see how we can address some of the pitfalls and challenges that need to be addressed in such analyses. And I'll also provide some conclusions at the end. So creatine plays a key role in energy metabolism, especially during short bursts of high-intensity activity. In the human body, it exists primarily as free creatine and also as phosphocreatine, which is serving as a rapid energy reservoir. When energy demand spikes, such as during the exercise, phosphocreatine donates its phosphate group to adenosine diphosphate and quickly regenerates adenosine triphosphate, which is the primary energy currency of cells in human body. Creatine is found naturally in the muscles and brain, as well as in other energy demanding tissues, and also and can be obtained either via diet, typically by consumption of red or fish meat, or via supplementation. And of course, it's also endogenly synthesized in the body. Creatine has seen a significant rise in popularity within the sports and fitness community, becoming one of the most widely used and research performance enhancing supplements. Its appeal spans across athletes, bodybuilders, and recreational fitness enthusiasts, owing to its broad range of benefits and versatility across many product types. At its core, creatine supports several key areas of physical performance and recovery. One of its most and well-established effects is enhancing performance in high-intensity, short-duration activities, such as sprinting or weightlifting. In addition to performance, creatine contributes to increased muscle mass and strength over time. It also supports faster recovery between workouts, allowing for more consistent training and reduced fatigue. Emerging evidence suggests that creatine may also improve aerobic capacity in some individuals, and its potential neuroprotective properties are currently being explored and studied in the context of brain health and cognitive function. So many proved and many potential bioactivities in human body. The growing interest in creatine is clearly reflected in market trends as well. In 2024, the US creatine supplements market alone was evaluated almost at $420 million. And this substantial figure highlights the continued and expanding demand for creatine as both a performance and wellness supplement. The market growth is largely driven by two key factors: rise in general fitness awareness across the population and the increasing acceptance and use of performance-enhancing supplements, not just among athletes, but also among recreational users and I would say health-conscious consumers. As creatine's benefits become more widely understood and supported by scientific evidence, this upward trend is expected to continue, as shown in this graph. Creatine monohydrate remains the benchmark form of creatine and is backed by decades of research and extensive use. It is well characterized in terms of physicochemical properties. It is highly stable in its powdered form and demonstrates excellent bioavailability. One of its most compelling advantages is its affordability, making it relatively accessible and cheap. Importantly, creatine monohydrate is supported by a large body of scientific literature and demonstrating its effectiveness across multiple performance and health-related outcomes. In recent years, there were numerous alternative forms of creatine. These have entered the market and were often accompanied by claims of enhanced solubility, absorption, or efficacy. However, these claims are typically not supported by a large amount of data. It's typically based on limited data, and in some cases, there's no data supporting these claims at all. The alternative forms that are used in supplement ingredients represent various creatine salts, such as creatine hydrochloride, citrate or nitrate, various complexes, or even derivatives that have chemically modified creatine structure, such as esters or alcohols. Let's take a look at creatinine. This compound is the main degradation product of creatine and also degradation product of phosphocreatine, which is present in the body. Creatine spontaneously converts to creatinine through a non-enzymatic intramolecular cyclization, and this occurs especially in aqueous solutions. This degradation is accelerated under acidic conditions at higher temperatures and over longer storage times. While creatine is stable in its dry powdered form, it readily degrades in solutions, making creatinine a common impurity or degradation product in creatine supplements. In the body, creatinine is excreted by the kidneys and serves as a clinical marker of kidney function. Testing for creatine and its main degradation product creatinine is crucial to confirm the identity and purity of the active ingredient that is formulated in dietary supplements. Creatinine serves as an important marker of product stability and shelf life, and accurate analysis ensures regulatory compliance and verifies that label claims are truthful and reliable. Beyond creatine and creatinine, it is equally important to test for a broad range of other parameters to ensure the product quality and safety. These include process-related impurities that may be formed during synthesis of creatine monohydrate, heavy metals, residual solvents, allergens, proteins, amino acids, vitamins, and other ingredients. And other potential targets for testing are adulterants, which can be used as adulterants. These would be performance boosters, different pharmaceuticals, and Last but definitely not least, it's important to test for microbial pathogens. Together, these tests, of course, help manufacturers to deliver safe, effective, and high-quality creatine supplements. Let's take a look at the typical analytical workflow used in the determination of creatine and creatinine. We have 3 stages here: sample preparation, separation detection, and quantification. When it comes to sample preparation, the sample is dissolved in water or buffer, and extraction is performed using agitation or sonication at ambient temperature. Alternatively, elevated temperature may be used as well. After that, the extract can be filtered and additional dilution may be performed to fit the analyte responses into calibrated range. After dilution, there may be additional filtration. When it comes to separation by high-performance liquid chromatography, there are different separation systems that can be used. These include reverse phase separation, occasionally used with ion pairing agents, ion exchange chromatography, helic chromatography, or mixed mode chromatography. And when it comes to detection, the most widespread detection would be UV or DAD detection at wavelength between 210 and 240 nanometers, and also alternative detectors can be used to boost sensitivity, selectivity of the determination. And quantification would be performed based on external calibration with calibration standards prepared in solvent. Okay, switching gears to challenges. Again, 3 different stages of the LC analysis of these compounds. When it comes to sample preparation, it is really important to take a representative sample size for the analysis and ensure we are aliquoting the sample from a homogeneous and representative sample. Another challenge is related to sufficient and complete dissolution of target analytes and sufficient dissolution or dispersion of sample matrix in the extraction medium. And finally, considering what we already talked about in terms of stability in solution, it is of paramount importance that your analytes are stable during the extraction, but not only during the extraction, but also over the time the sample extracts and standards spent on the instrument. And of course, for the instrumental analysis, it is important to achieve appropriate and sufficient resolution between creatine and creatinine, but more importantly, it is important to make sure that there are no coelution with matrix components or other interferences you may see in your chromatographic record that could hinder or interfere with the accurate quantitation of the targets. And when it comes to quantification, there are certain challenges that are associated with quantification of different creatine forms we talked about previously. So looking at our internal method, our method is applicable to water-soluble forms of creatine that do dissociate into free creatine. The matrix scope of our method are dietary supplements and beverages. And our method was validated down to the limits of quantification that are displayed in this figure. You can see an example chromatogram we typically obtain with our analysis with creatine and creatinine peaks well resolved. In this particular record, you can see an overlay of the lowest calibration standard and highest calibration standard represented by red and black traces respectively. So looking at the sample preparation workflow, it's pretty much aligned with what I outlined previously. So it's typical sample preparation workflow in this arena. We take a representative sample, at least 5 grams, into a volumetric flask, and then we fill the flask with ultra pure water, not full, not completely full, 3/4 full. And then we perform extraction on a horizontal shaker, and we do extract at ambient temperature. So the default extraction time in our method is 20 minutes, but we, based on our validation data, we can prolong the extraction time up to 60 minutes, especially for samples that are challenging in terms of dissolution. And also we can support the extraction process by brief sonication of the extraction mixture. Typically we use times below 3 minutes. So after that, the volumetric flasks are brought to volume again with water and vigorously shaken just to make sure everything's homogeneous. And after that, depending on the expected or previously determined concentration of creatine in the sample, we perform dilutions. With water, we take a filter, an aliquot of the sample, filter it through a PVDF filter, violate and analyze by HPLC-UV. So in our method, we are using C18 column with polar end capping. The mobile phase is composed of water that is buffered to pH of approximately 6. Elution mode is isocratic, and we are able to complete sample analysis within 7 minutes. And we detect at detector wavelength of 210 nanometers, and our samples and standards are kept at 5 degrees centigrade. Let's take a look at the quantification approach. As I said, we are able to analyze water-soluble creatine forms that dissociate into free creatine. And our quantification approach is based on free creatine, basically. So we always work with anhydrous creatine reference standards, which we store at ambient temperature in a desiccator. We've heard that creatine is pretty stable, and we can just confirm that. But although this standard is anhydrous and stored in a desiccator to prevent moisture, or to avoid capturing moisture, we also incubate an aliquot of the reference standard at 100 degrees Celsius for 1 hour, and this is used to completely eliminate any residual moisture that might be caught by the material during the storage. Then we prepare the calibration standards in water, and using a linear calibration curve, we quantify free creatine that is present in the sample using those solvent-based standards. And once we determine the actual content of free creatine, we recalculate to respective forms using appropriate conversion factors. So in this table, you can see which different creatine forms we are able to cover with our method, and also the respective coverage factors. Of course, during the validation of our method, we also wanted to demonstrate that all these creatine forms actually do dissolve under the conditions of the method. So let me show some of these results. In this slide, you can see records for anhydrous creatine and free creatine salts that are covered by our method. So the way we worked, we dissolved the reference standards under the conditions of the method, and the concentration was normalized to 10 micrograms per milliliter, expressed as creatine anhydrous. So there was a good promise that all these forms will dissolve completely, just based on the information available in the literature on solubility in water, as you can see in this table. And comparing the normalized peak areas obtained for respective solutions, we see that our quantification approach is valid because all these salts do perform or do dissolve completely under the conditions of the method. We are still looking at creatine nitrate. It's somewhat difficult to obtain sufficient amount of this standard, but again, based on solubility, we do expect to see sufficient dissolution. There are some limitations that are associated with the applicability of our method. So in addition to those water-soluble salts, we also obtain a standard of a derivative, creatine molecule esterified with ethyl, creatine ethyl ester. And you can see when we compare the chromatogram of creatine and creatine ethyl ester, which is one of the forms that have been used for final product formulation, we see that the method gives completely different retention time. So, you know, the approach I outlined previously is not applicable to forms that include chemically modified creatine structures. Let's take a look at the method validation. Method validation is crucial because it demonstrates that the actual method is suitable for its intended use, and it's also a requirement in order to get the method ISO accredited, and also provides a lot of really important and interesting data on the performance of the method. Let's go through the different method validation parameters we evaluated. So we looked at selectivity. We ran a method procedural blank sample that's carried through the sample preparation but contains no matrix. We ran some blank unspiked matrices. We evaluated potential carryover from injection to injection. And we also performed identification of the analyte in the samples based on comparison with the standards. We also looked at linearity of the response. That's crucial for quantification. And we evaluated accuracy and precision. This was performed with two different sets of samples. We had blank matrices that were fortified with both analytes. And we also analyzed actual finished products that were formulated with creatine monohydrate. And actually, creatine monohydrate was the form we focused on as it is most widespread. And you know, there are only limited samples on the market that would be formulated with other forms of creatine. And of course, for accuracy and precision, these experiments were performed across multiple days by at least two different analysts. We also looked at robustness, evaluating prolonged extraction time, and also stability of both extracts and standards. And also we took a look at any potential chemical changes of the anhydrous creatine after the drying process. The LOQs of the method were demonstrated based on evaluation of the results obtained for the lowest fortification levels that were performed in the validation study. Regarding acceptance criteria, we didn't want to see analyte signals in our method procedural blanks. We didn't want to see any carryover, so this blank sample was actually injected after the highest calibration standard, just to see that we are not introducing any analytes from high concentration injection to the following concentration. And then for analyte identification, our acceptance criterion was pretty narrow. It had to be within 0.05 minutes of the average value obtained or observed in the standards analyzed in the same measurement sequence. Pretty typical requirements for linearity, coefficient of determination of at least 0.995, and residuals within 5 and 3% at the lowest and other levels respectively. Our accuracy and precision criteria were adopted from official methods of analysis of AOAC International Appendix K, guidelines for dietary supplements and botanicals. These are concentration-dependent criteria. This table shows an overview of acceptable recoveries depending on different analyte concentration or concentrations in the sample. So we use that to evaluate our validation data. When it comes to precision, we used values from modified Horowitz equation, again aligned with the AOAC International guidelines. And this table again shows what were our acceptable RSD-R values, which would be repeatability values, and RSD-IMT, which would be intermediate precision values. The RSD values are obtained by single analyst on a single day, while the relative standard deviations under intermediate precision conditions are calculated based on data collected on different days by different analysts. And as you can see, these are again concentration dependent. The lower the concentration, the higher the acceptable RSD, and vice versa. As I mentioned already, we had two different sets of validation samples. So we had blank samples and we had actual samples formulated with creatine monohydrate. And this slide shows an overview of ingredients that were included in the actual matrices. We included blank protein powder, non-protein powder, which would be typically pre-workout powder with different ingredients. We included gummies and beverages. And in case of formulated products, the actual creatine monohydrate values range from approximately 6.7 up to 100% for actual pure creatine monohydrate powder. Experimental design. Blank matrices were fortified with both creatine and creatinine at least at 2 levels, one of which would be at or below LOQ of our method, and the highest one around 25 and 12.5% by weight for creatine and creatinine respectively. For formulated products, we also looked at the sample size. So we worked with either 1 gram or 5 grams. The sample to extraction volume ratio was maintained the same, but we explored or wanted to see what the impact of the sample size is on the results. And all the samples were analyzed on 3 different days by 2 different analysts, resulting in a total 9 replicates for spiked matrices across 3 days. And in addition to these validation samples, we also validated our laboratory control sample by analyzing over 10 different days by 2 different analysts to establish acceptable values. This sample would be later on analyzed with our routine samples just to control the process, and would be control charted to look for any trends in results. Okay, showing some results for selectivity, just to demonstrate that our chromatographic records are pretty clean and free of interferences. You can see an overlay of method blank and lowest standard, and then 2 example matrices of blank protein powder and powder spiked at LOQ, the same record for gummies, and then also chromatogram we typically obtain for pure creatine powder, You can see there's a single peak. It's pretty boring chromatogram, but that's the way we like it. One thing I didn't mention is that none of the formulated products that were included in the validation contained any significant amounts of creatine that we could either see values really well below the target LOQ or none at all. So selectivity was deemed acceptable. We didn't see any carryover as well, and our matrix blanks were nice and clean. This is a summary of spike recovery results for 4 of the blank matrices that were fortified at different levels, and the black rectangles in the graphs actually show the acceptable range for mean recovery per AOAC guidelines. You can see that in most of the samples we are really close to 100% at both low and high level for both analytes. Just to mention, creatine values would be the values in blue, while the creatinine are in gray. So pretty good results, definitely within acceptable range per AOAC guidelines. And also wanted to mention that the actual RSDs were acceptable, meeting the acceptance criteria I showed you previously. Here are some results for formulated products, and also in comparison of mean concentrations of creatine monohydrate determined in these matrices with 1 and 5 gram sample size. So clearly you can see that if we are taking higher sample aliquot for the analysis, we are kind of getting closer to the label claim. That's what we did. We also looked at the label claim of the particular product, and 5 gram results interestingly showed results closer to the label claim provided by respective manufacturer of the product. Interestingly, in chewables and gummy products, we could see relatively higher concentration of creatine. That it was kind of interesting. It was formulated over what was claimed, which may have different reasons, and we may discuss this a bit more later on in the presentation. In any case, we didn't see any creatinine results, which is good and shows good quality of these products along values that comply with the label. And in this graph, we do plot actual RSDs obtained under intermediate precision conditions, and we are comparing results for different sample sizes in case of protein or protein powder and pure creatine product. And you can see overall we are getting better RSDs with larger sample size. It's important to take larger sample size based on our experience and validation results. In case of protein powder product, this is major improvement in precision depending on the sample size you use. So higher sample size is important to address potential inhomogeneity and representativeness of the sample. The next parameter we evaluated was the stability. So I'm not showing it in this actual presentation, but we didn't see any degradation of the anhydrous creatine that is actually heated prior to preparation of the calibration standards, which is good and was expected based on information in the literature. Higher concentration standards, which would be our stock solution and intermediate standards, we do prepare from lead materials prior to the preparation of the samples, and calibration standards always on the day of analysis, just to avoid any stability concerns or potential impact on the analysis. But we did evaluate the stability of sample extracts, and we picked 2 matrices for this purpose. These would be the pure creatine monohydrate powder and also non-protein powder fortified at approximately 25% with creatine, and creatinine at half of that concentration. And you can see when we store the samples for 24 hours at approximately 5 degrees Celsius, which would be the temperature maintained in the HPLC-UV instrument autosampler, we do see acceptable and really minor changes. And this data actually shows that we are demonstrating stability, and we are confident we don't see any degradation of the target analyte or target analytes in the sample over this period, which would be sufficient to complete the measurements in the samples. And the same applies to working standards. So we did evaluate the lowest working standard and the highest working standard. And again, the relative percent difference stands for RPD in this table, where within our acceptance criteria and showing good stability. And as mentioned, we also looked at robustness. The reason we looked at extraction time was just that it's really useful to be able to extend the extraction time in some cases and for certain samples. So we performed this analysis for formulated protein powder and pure creatine monohydrate powder, and we performed extractions per the method per our default extraction time, that would be 20 minutes, and we prolonged the extraction to 40 and 60 minutes to see what would be the impact on the concentration of creatine in that particular sample. We can see the relative percent differences from 20-minute extraction are pretty low and within the variance of the determination, I would say, in both matrices. So this gives us flexibility in case we can visually see there are issues with dissolution of the product at 20 minutes extraction time. We can prolong it and be confident that we don't see degradation of our target analyte over that time. And this slide is just to show you some system suitability and routine quality control measures we are introducing. So system suitability is basically a set of requirements that ensures the instrument performance is adequate for the method. So we would always inject a set of standards prior to submitting the full sequence to confirm that the peak shape of our analytes are within acceptance criteria and look good, just to make sure that maybe the chromatographic column is performing well and it's not deteriorating. And also we are checking stability of the retention time. So the person RSD, and I'm sorry it's missing the information, the RSD calculated for retention times in calibration standards must be below 2%. And then additional quality control measures, we always analyze method blank, and it needs to be free or well below the LOQ in terms of analyte response. I already mentioned the laboratory control samples, so these values were validated for creatine because the material contained creatine, so we established mean value that's acceptable and also margins within which the actual results obtained in respective routine batches should be. The results in this laboratory control sample is charted and evaluated for trends, which gives us additional confidence in the results. And the same laboratory control sample, which did not contain any creatine, therefore we couldn't establish except mean or assigned value to be monitored further on. This LCS is also fortified with creatine, so we can monitor spike recovery for this analyte. And again, we can control chart these values and look for trends and any aberrant data. One additional issue I wanted to highlight, when I was preparing this presentation, I saw some really interesting information when it comes to creatine gummies and analysis of this matrix. So Now Foods in 2024, they performed a really limited market survey that focused on creatine content in this matrix. So they sourced 12 different brands of samples, which represented single lots for each product, and they analyzed it with their internal validated method. And interestingly, half of the tested products failed the label claim. And in the table on the right, you can see that in some cases, these were major. There was a major discrepancy between the label claim and the determined results. Sometimes not seeing any creatine at all, with some relatively low levels of creatinine that indicates that there could be degradation of the sample of creatine in that matrix. So pretty interesting results. And the question is, was the testing method fit for purpose? Well, it seemed to be validated, so probably yes. So could this be caused by homogeneity of the sample, or some problems with the product formulation or stability issue? So just something I wanted to highlight, that this particle matrix may be difficult in many ways. So what we did, we also randomly sourced 6 different gummies, and all of these were well within the expiration date, and we performed analysis with our method, and surprisingly we saw similar rate of failures in our tests. So sometimes there was no creatine at all, or even no creatinine. You may question the method, but finally, ultimately, the method dissolves the whole gummy. So the whole matrix, which is prepared by liquid nitrogen homogenization of the full pack, is dissolved in water. So we should see at least something. So these observations are, again, really interesting. And I was trying to dig into why this could be, and it seems that formulation of creatine gummies is pretty challenging. So obviously, there's high temperature involved in production of gummies. So seems that the actual temperature or stage at which creatine monohydrate is added into the formulation may play a crucial role in the outcome, whether you see really low concentration in the final product, or you're at or above the label claim. So let me conclude by saying that obviously creatine and also creatinine testing is essential to ensure the product compliance. I would strongly recommend to test stability in your finished product, not only at the beginning of the shelf life, but also towards the end of the shelf life, just to show and be confident that the concentration of active ingredient is stable or still at or above label claim throughout the shelf life of the product. I showed you our internal method with some validation data. There's more validation data I could show, but we have limited time. So what I showed hopefully convince you about acceptable performance of the method when comparing to AOAC International performance criteria. And just a note on new complex samples. So currently the method is applicable to only really water-soluble matrices, or matrices compatible with water, but there are additional forms of supplements or foods, even pet food, that's formulated with creatine. So looking at such matrices that definitely would require some modifications to the extraction procedure, and our method as such would not be applicable to these matrices. I would like to acknowledge my colleagues at Eurofins Food Chemistry Testing, Madison, Wisconsin, especially Lauren Kelly, who did most of the validation work, but also other members of the team. And I would like to thank you for your attention. I'll be happy to take any questions. Thank you.
Okay, perfect. Thank you, Lukasz. That was a very excellent, very informative presentation. So we have a couple of questions in the chat. So the first one, what would you suggest for next steps if we are getting lower than expected creatine results in a gummy product?
Typically when we get out of, we do obtain or report out of specification result, we would recommend retesting. So we can do that. We do multiple retests, and then we can evaluate the results, whether it confirms or it kind of shows the results are inconsistent. If it confirms, I would recommend to look into the shelf life, maybe send a fresh sample just to confirm again. But you know, if it fails your expected results, it may be associated, especially in the case of gummy matrix, maybe associated with formulation or stability of the product. So would likely recommend to look into that, and obviously we can support you in such investigation from analytical perspective.
All right, perfect, thank you. Another question we have, are there plans to expand the method to more complex products such as bars or food?
Yeah, that's something I mentioned. We haven't started the method development or method optimization yet. That said, we do see increased requests from our clients when it comes to testing in nutritional bars. So that's something we may start looking into. We already have some ideas how to tackle this matrix, but as for now, we are not actively looking into this, but it's in the method development pipeline of our team, I would say.
Super, super, super. Well, thank you so much, Lukasz, for today's presentation. And we also just want to say thank you to everyone who's attended. Please feel free to reach out with any questions, and we would be happy to connect further to discuss this important topic. This now concludes today's presentation. Have a wonderful day.